instinctively not using criteria that were scientific but born out of a need to use organism for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom classification system with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic, that is green algae, and non-photosynthetic, that is fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, a two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationship, etc. Classification systems for the living organism have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristics of the five kingdoms. Characters: cell type, prokaryotic in Monera, eukaryotic in Protista, fungi, Plantae, and Animalia. Cell wall: non-cellulosic, polysaccharide plus amino acid in Monera, present in some in Protista, present with chitin in fungi, present with cellulose in Plantae, and absent in Animalia. Nuclear membrane: absent in Monera and present in Protista, fungi, Plantae, and Animalia. Body organization: cellular in Monera and Protista, and multicellular or loose tissue in fungi, tissue or organ in Plantae, and tissue, organ, or organ system in Animalia. Mode of nutrition: autotrophic, chemosynthetic, and photosynthetic, and heterotrophic, saprophytic, and parasitic in Monera, autotrophic, photosynthetic, and heterotrophic in Protista, heterotrophic, saprophytic or parasitic in fungi, autotrophic, photosynthetic in Plantae, and heterotrophic, holozoic or saprophytic in Animalia. R.H. Whittaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. Let us look at this five-kingdom classification to understand the issues and considerations that influence the classification system. Earlier, classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones, say, for example,